Once a bacterial pathogen has crossed into the human body and attached to its tissues, that pathogen begins to reproduce. But how does it exert its effects and cause disease? Unfortunately, we don't have time to get into the nitty-gritty details of how bacteria make us sick, but we can talk about a few of the ways. In this video, we'll briefly talk about how pathogens generally and then bacteria specifically cause disease. We'll talk about siderophores, the iron-stealing molecules, and exotoxins, proteins that disrupt cellular functions in a host. By the end of this video, you should be able to explain the role of siderophores during an infection, and you should be able to distinguish among the exotoxins in terms of naming, how they're produced, what they are, their functions, and the diseases that they cause.
So we're in chapter 15, and so far we've talked about ways that pathogens cause infection. So we've talked about some of the factors involved. Remember, we talked about the portals of entry. We talked a little bit about the number of microbes and how we can, in a laboratory, kind of compare and determine how many microbes are necessary for an infection. We've talked a little bit about the lethality and how we measure that in a laboratory and how we can use that to compare the different toxins that pathogens produce. We've talked about the mechanisms that they use to penetrate deep into the body, to get past the skin. So in the second half of chapter 15, what we want to do is actually look at what pathogens do once they actually get into your body. Then they've penetrated your defenses. They've made it past the phagocytic cells, like your neutrophils and your macrophages, and the bacteria are kind of in a place where they can actively grow. So it turns out there are 4 ways that pathogens hurt their hosts. The first thing that they might do is they'll use host nutrients. The second thing that they'll do is they'll cause direct damage at the location of infection or their entry point. The third thing that they might do is produce toxins. And then the fourth thing that they do is produce a kind of special hypersensitivity reaction. And before I finish today's lecture, we will have gone through all of these different ways or methods of damage. So you'll recall that microorganisms, aren't necessarily harmful, just generally speaking. But in this chapter, we're considering pathogens. So pathogens are disease-causing microorganisms. And like all other living things, they have certain requirements for their growth. And one of those requirements is going to be nutrients. They need lipids. They need carbohydrates. They need proteins. All of these things to kind of support their growth. In addition to lipids, carbohydrates, proteins, and nucleic acids, actually, microorganisms, especially pathogens, are going to have trace elements that they use. So one element that's really important for certain microbes to live is called a siderophore. A siderophore. Now these are small, low molecular weight molecules. You can see a picture of one actually on this slide. And what these do is they bind iron in the environment. So we often find bacteria and fungi that are under low iron stress. That is, there's not enough iron really to support their growth. They'll produce these molecules and they'll secrete them. And these molecules grab free iron in the environment, and then the bacteria and the fungi can reabsorb that siderophore and the iron that's bound to it. So you might be wondering, well, why do they need iron? Well, it turns out that if a bacterium or a fungus is doing any kind of aerobic cellular respiration, they need iron in order to reduce oxygen. So oxygen, you'll recall, is essential for aerobes and your facultative anaerobes. And these types of cells, well, they need iron to reduce their oxygen. In addition, you have instances where some bacteria and some fungi will use iron as part of their process for making the precursors for DNA. So without iron, these cells can't make DNA and synthesize it, which means they can't reproduce. Still, other cells need to make heme, and heme requires iron. So we'll find that some microbes, some pathogens, need these siderophores to try to recover iron from the environment to carry out these processes. Now, here's the thing. Most iron is already bound to some sort of protein when we consider a host. In mammalian cells, for example, you have these iron transporting proteins like transferrin, lactoferrin, and ferritin. Transferrin kind of serves a protective function in the human body. This protein has a very strong affinity or attraction for iron. So as it circulates in the blood, what it does is it grabs any free iron that's around, and then since that iron is not available for bacteria and fungi, it helps keep those bacterial cells and those fungal cells from growing. That might be in the blood, which is unusual, by the way. Lactoferrin is another iron-binding protein that's made by host organisms, and it's similar to transferrin. We find lactoferrin in secreted fluids, kind of like sweat, tears, and milk, and it's going to bind any free iron, again, preventing that iron from being absorbed by pathogens. So again, there's a little bit of a protective function. Ferritin does kind of the same thing, but it's actually inside the cytoplasm of a cell. So again, the idea here is that most of the free iron inside of a host is going to be bound by some sort of protein, and that makes the iron unavailable for microorganisms that need that iron to support their growth. So what some bacteria do is they make these siderophores. So just in case there happens to be any free iron around, the siderophores can bind that iron, and then it's absorbed by the bacterium. There are some instances where siderophores will also steal iron from some of these iron-binding proteins. Pathogenic microorganisms can also cause damage directly. So remember we said that pathogens can enter the body, and they'll usually come in through a preferred portal of entry because it's close to the tissue that they infect. Well, once those microbes land on that tissue that they infect, they'll attach and they'll begin to grow. Now, as these pathogens are growing, of course, they're going to be absorbing nutrients from their host, okay? But in addition, they'll start producing waste products. So as they're metabolizing the host's nutrients, the proteins, the lipids, carbohydrates, etc., they'll produce waste products. And some of these waste products can be dangerous. They can be actually harmful to the cells in the tissues that these microbes are growing on. In addition, sometimes the infection of host cells by pathogens can actually lead to rupture of those host cells, which again leads to tissue damage. Okay, and then you know from this chapter that some bacteria in particular are able to penetrate host cells, and as they do that, they can actually damage those cells. So in these ways, stealing nutrients, producing waste products that are harmful, rupturing cells, and penetrating cells, pathogens can actually damage and cause direct damage, rather, to host tissues. Still another way that pathogens can cause direct damage is by producing toxins. So we want to spend a little bit of time talking about toxins. A toxin is a poisonous substance that's produced by a pathogen. Not all microorganisms will produce toxins, but many pathogens do. When a pathogen produces a toxin, that's usually the primary factor that contributes to its ability to cause disease. When a pathogen is capable of making a toxin, we say it has toxogenicity. And when that toxin makes it into the blood of a potential host, then we say that that host has toxemia. Now, people can be vaccinated against toxins. The inactivated toxin that's used as a vaccine is called a toxoid. So you may be familiar with the Tdap vaccine, which immunizes people against tetanus, diphtheria, and pertussis. So for some formulations of this, a toxoid for tetaноspasmin, this is the toxin that tetanus, Clostridium tetani, the bacterium that causes tetanus, makes. So what happens is you inject an inactivated form of tetaноspasmin into a susceptible potential host for the bacterium. And then if that person becomes infected by tetanus, Clostridium tetani, and the bacterium starts making the toxin, then that person has the ability to make antibodies against the toxin, and thus they're protected. The actual antibodies against the toxoid are called antitoxins. So you've probably heard of antitoxins before. They can actually be purified and administered as a therapeutic in the case where someone is actually suffering from the effects of a toxin. When we consider bacteria, there are two main types of toxins that you need to be aware of. The first type of toxin is called an exotoxin. Exotoxins are encoded by genes within the bacterium. They're synthesized within the bacterium, and then they're secreted into the environment. Now, if we're talking about a bacterium that's inside of a host organism, that toxin can be absorbed by into the interstitial fluid, and then it can be absorbed into the blood. And once it's in the blood, it can be transported throughout the body where it might have an effect. In addition, these toxins can make it into the lymph system. And you'll remember your lymphatic vessels run all over the body, so that means that the toxin can be carried throughout the body as well. Your second type of toxin is called an endotoxin. Endotoxins are actually part of the cellular structure for some bacteria. When these bacteria die and the cell starts to break down as a result, this endotoxin gets released, and then it will exert toxic effects inside of a host. So the main thing you need to remember is that exotoxins are secreted into the environment. They are not part of the bacterial cell, but rather they're made as part of the life process of a bacterium. Endotoxins, on the other hand, are part of the bacterial cell. And it's once that bacterial cell dies and starts to break apart that endotoxins actually have an effect. So for the next few minutes, what I want to do is focus on exotoxins specifically, and then endotoxins, so that you can be more familiar with what they are, what kind of effects they can have, and kind of their significance with respect to pathogenicity. So we're starting with exotoxins. And one thing you want to know about exotoxins that I've already mentioned is that in order for a bacterium to make an exotoxin, they have to have a gene to make it. That gene can be part of the bacterial chromosome, or it can be imported on a plasmid or via a virus. Most exotoxins, in fact, probably all of them, are protein-based. They're often enzymes that can act over and over and over again. They can carry out the same reaction over and over and over again in the body. Oftentimes they're going to disrupt some sort of natural function inside of a host cell, or they will disrupt cellular structure for a host. Now, because these are enzymes that can act over and over and over again, they can carry out the same chemical reaction several times, it means that minute quantities of exotoxin can actually exert a large effect inside of a host. So that disruption that I was talking about can happen many, many times just due to a nanogram, for example, of toxin. With respect to bacteria that make exotoxins, it's the exotoxin that the bacteria make that causes the disease, not the actual bacterium. So exotoxins are named based upon 3 different criteria. Some are named for the type of cell that's attacked. So for example, a cardiotoxin would attack what kind of cell? Right, heart cell. And an enterotoxin would attack what kind of cell? I hope you remember this. We said entero refers to the intestine, so these would be intestinal cells. And your exotoxin can also be named based upon the disease that they cause. So for example, Corynebacterium diphtheriae produces diphtheria toxin, and that's responsible for diphtheria. Diphtheria is a disease where a thick covering forms at the back of the throat, and it can cause difficulty breathing. In severe cases, it can actually lead to death due to suffocation. Tetanospasmin causes tetanus, this condition where the skeletal muscles of the body the skeletal muscles of the body are constantly spasming. You've probably heard of lockjaw before. This is one of the first symptoms of a tetanus infection. So here, the masseter, which you'll remember are these large muscles that help control the jaw, they start to spasm, and the spasming of the masseter muscles actually freezes the jaw in place. That spasming will spread from the jaw throughout the body and can lead eventually to respiratory arrest and death. Sometimes exotoxins are named for the bacterium that produces them. So, for example, we've talked about Clostridium botulinum. This is a bacterium that produces the botulinum toxin, and the botulinum toxin is one of the most toxic toxins, most poisonous toxins known to man. Only a few nanograms can kill a person. So remember that here, the toxin instead of causing spasming in your skeletal muscles, it causes relaxation, so flaccid paralysis. This can affect your breathing because you can no longer contract muscles like the diaphragm and your intercostals, which help with lung function. Vibrio cholerae produces Vibrio enterotoxin. So this is a toxin that attacks the intestinal intestinal cells, causes them to release ions into the lumen of the intestines. That's followed by water, which leads to diarrhea. But it's named for Vibrio cholerae, the bacterium that produces the toxin. When we talk about exotoxins, we can group them into 3 groups according to their functions or activities. The first group are called the A-B exotoxins, and they're illustrated on the next few slides. So if we look at figure 1 here, what we're looking at is a bacterial cell. You can see the bacterial chromosome represented by the circle at the top, and on that chromosome we would have a gene for producing this type of exotoxin. That gene gets transcribed by RNA polymerase, producing an exotoxin messenger RNA. When that messenger RNA is translated by the cellular ribosomes from the host, we get a 2-part toxin. That's the AB exotoxin. So it's represented by that little, it looks kind of like a man with a head and 2 legs in this picture. So the A in this AB exotoxin stands for active. This is the actual toxin itself. The B stands for binding. So this is a second part of the protein. And what it does is it delivers part A to cells that can be affected. In part B, or number 2, here we can see that the AB toxin has been secreted from the bacterial cell and now it's binding to a host cell. So this is a cell within a tissue inside of a host, and its function is about to be disrupted by this toxin. So notice that the B portion of the toxin binds a specific receptor on the host cell. This is how the toxin is able to be targeted to the cells that it can affect. So part B of the AB toxin binds the host receptor. This triggers endocytosis of the toxin. So it comes into the cell in this little vesicle. So here, our AB toxin has been enclosed completely in a vesicle, and it's in the cytoplasm of the host cell. Inside of that vesicle, the A and the B components of the exotoxin are going to separate. The B portion gets recycled. It's transported back to the surface of the cell, and it's exocytosed and released basically into the interstitial fluid. The A toxin, on the other hand, the A portion, is going to inhibit protein synthesis. So if a cell is unable to make the proper proteins, what happens to that cell? Right, it becomes damaged and potentially dies. The death of several cells within a tissue can lead to things like organ failure. This is one way that bacteria can cause direct damage to host cell tissues. So this is a table from your textbook. It's table 15.2, and there are three diseases that I've starred here. You should be familiar with the name of the disease, the bacterium that causes it, and the mechanism. So what does the toxin actually do from these bacteria? So you'll want to know that for your exam. Another way that exotoxins can be harmful to a host is by lysing the cells of the host. So remember that inside of a host, think about ourselves, for example. We have these wonderful immune systems that are full of cells designed to bind and destroy invading cells like bacteria and fungi, or non-living particles like viruses. So these membrane disrupting exotoxins are going to do one of a couple of things. They may form channels or holes in the plasma membrane of protective cells like neutrophils and phagocytes, causing them to lyse, or they may just disrupt the phospholipid bilayer, which also leads to lysis. So there's a couple of things that can happen here. The common thread is that these types of toxins are going to target cells and cause them to break open. Now, your membrane disrupting toxins can be classified into kind of three groups that I'm interested in you knowing. You have the leukocidins. These target white blood cells. So these are the ones that are going to target your neutrophils and your macrophages. They're going to punch a hole in the membrane, cause those cells to be destroyed so that they can't go around phagocytizing more bacteria. In addition, these leukocidins can release bacteria that have already been phagocytized and are enclosed in phagosomes. So these will punch holes in those phagosomes, cause them to break open, releasing bacteria. You also have a class of proteins called hemolysins. Hemolysins break open red blood cells. Why would bacteria want to do this? Well, remember a little while ago when I told you that some bacteria need iron? Remember that red blood cells have hemoglobin in them, which binds iron. Siderophores can steal iron away from hemoglobin. So releasing hemoglobin by breaking open red blood cells actually benefits those bacteria. Leukocytins and hemolysins are both produced by staphylococcal cells like Staphylococcus aureus and by streptococcal cells. Streptolysins, this is a third group of membrane disrupting exotoxin, but it's only produced by streptococci. And your streptolysins are actually a class of hemolysin. This is another part of table 15.2 that gives you a couple of examples of membrane disrupting toxins. I'd like for you to be familiar with the Clostridium difficile toxin, which is a membrane disrupting toxin. So please, I forgot to put a star on this slide, I'm sorry, but superantigens are bacterial proteins that trigger an intense immune response. This is that hypersensitivity response that I mentioned earlier in the lecture. What these proteins do is they stimulate the proliferation of a specific type of immune system cell called a T cell. T cells are kind of like the generals of the immune system. They're going to help detect and target specific invading cells in a host body, and they're going to help control the immune response to those specific cells. Now, under the control of superantigen, these T cells become many in number, and they release an enormous amount of a chemical messenger called a cytokine. So if you imagine an empty room and there are 10 people in the room talking, you as a listener might be able to focus on the conversations of a few people in the room at one time. Under the effects of a superantigen, it's like instead of having just 10 people sending messages, you now have 100 people sending messages. It would completely overwhelm the listener in such an example. So here, the human body becomes overwhelmed by the release of so many chemical messengers from T cells. The result is that the cytokines, these messengers, enter the blood, and they can trigger a variety of unpleasant symptoms like fever, nausea, vomiting, diarrhea, and in severe cases, shock, which is a life-threatening decrease in blood pressure, and superantigens can also lead to death, primarily because of shock. You might have heard of septic shock. This is a type of shock caused by bacteria. This is sometimes caused by these superantigens. This is another part of the table from table 15.2, and it shows you a couple of bacteria that produce superantigens. So for example, Staphylococcus aureus produces an enterotoxin, which is a superantigen, and it causes the secretion of fluid and electrolytes into the intestines, and that results in diarrhea. You also have toxic shock syndrome, which is produced by Staph aureus. It's caused by a toxin that's secreted, and it causes the secretion of fluids and electrolytes from capillaries this time. And that decrease in blood volume lowers the blood pressure, and it can actually be quite harmful to a potential host. I would like for you to be aware of toxic shock syndrome. So you should memorize this information about this particular disease. I've put a star by it on this figure. There's one more type of toxin, and I don't really have a slide for this, but this is another type of exotoxin. It's called a genotoxin, and it's produced by certain gram-negative bacteria. For example, Helicobacter pylori can produce it. And this toxin damages DNA. It causes mutations in DNA, and this disrupts the cell, and particularly this cell cycle. Your genotoxins can actually result in cancer. So this is another type of toxin. So just to summarize, your exotoxins are produced from a gene inside of a bacterial cell. And then this is usually a protein. That protein is secreted into the environment where it can be carried via lymph or blood to various places in the body, and it can target tissues that are actually located far away from where the bacterium producing the toxin is in the body. Your exotoxins are produced mostly by gram-positive bacteria, but some gram-negatives do produce exotoxins. These exotoxins are proteins, so as proteins, this means they can be denatured, which is how we can make some toxoids. Your exotoxins don't generally cause fever. They can be neutralized by antitoxins. Remember, antitoxins are antibodies to toxins. Okay, and the LD50 for this type of toxin is very small. That means you don't need a lot of this toxin to cause disease or to cause death, really. Remember, botulinum toxin is an example of an exotoxin, and you need only a few nanograms to kill a human being. This slide is here to remind you that some exotoxins are actually transmitted from one bacterium to another via viruses, so via transduction. In this slide, you can see several bacteria on the left. The toxin that they produce is kind of in the middle column, and then lysogeny is on the right. So remember what lysogeny is? This is when you have a bacterial virus called a bacteriophage. It infects a host cell, and it actually becomes part of that host cell, at least until the cell becomes stressed and starts dying. Then the virus might excite itself. But if the virus is carrying that gene and it infects a bacterium, becomes part of the bacterial chromosome, then that gene for making the toxin becomes part of the chromosome as well. That's lysogeny. So the diphtheria toxin gene, for example, is transmitted via lysogeny. The botulinum toxin gene, also carried on a virus, it is transmitted during lysogeny. The cholera toxin that we talked about, it is transmitted via a virus, and we talked about this in class. It's transmitted via a virus. As long as the bacterium is infected by that virus, it has the ability to produce the toxin and cause diarrhea.
So now I hope you know that siderophores are molecules that are produced by some bacteria during an infection, and the general role of these siderophores is to steal iron from the host in order to drive the growth of the pathogen, the bacterium in this case. We also talked about the different types of exotoxins. We talked about AB toxins. We talked about the leukocidins, and we talked about a number of other different types of exotoxins. Remember, exotoxins are basically proteins, and they're a byproduct of bacterial reproduction. As such, that means that these toxins can be denatured potentially using heat. We also talked a little bit about how they target specific cellular functions and cause disease. We also talked a little bit about the diseases that they caused. In the next video, we're going to talk a bit about endotoxins. Now, these are different from the exotoxins because while exotoxins are primarily made by gram-positives, but they can be made by gram-negatives, endotoxins are only found in gram-negative bacteria. I look forward to seeing you at that video.